All right, so this is going to be a bit of a maybe abrupt transition from matrices to higher order tensors, and also to a bit more of, I guess, a neuroscience-focused research talk rather than a general-purpose pedagogical talk. So at the outset, I want to thank all my collaborators, so especially Tony Kim from Mark Schnitzer's lab, who was really helped me work on this in the early days when I had this sort of crazy idea to apply tensor decompositions to neural data. And more recently, I've been working with Saurabh Vyas from Krishna Shenoy's group, who provided a data set in primate motor control. And so my two advisors, Tammy Kolda was really the one who helped me start this project. She works at Sandia National Labs, and Surya is my advisor at Stanford, who's sort of kept me going on it. Yeah. So the motivation for this project is that there's a lot of really exciting new experimental technologies and sets that neuroscientists are collecting. And this is one that really illustrates the point nicely. Here, this is from a lab at UC San Diego, and they're recording using calcium imaging in motor cortex as a mouse is learning a very simple lever pressing task. And here are basically raster plots on 3 different days as the mouse learns this task. And the red arrow denotes a lever press. So you can see that after many sessions, the mouse is able to press the lever in a much more stereotyped fashion on command. And this produces a very stereotyped pattern of neural activity within motor cortex. But on the first day, there isn't really this nice ordered representation in motor cortex. And what's sort of I love about this figure is that it really shows you that not only do we have the ability to record these like second level, sub-second dynamics that encode every individual lever press, but we can look at this same circuit over many days or even weeks as this sort of structure emerges. So really, like, neural data is getting very large, and that's something that people have been talking about for a while. But usually when we say we're entering the era of big data in neuroscience, they talk about the fact that we're recording from many neurons. But there's sort of multiple ways in which our data is really complex and high dimensional. So we don't only just record from thousands of neurons using technologies like NeuroPixel or large-scale calcium imaging, but we also record over thousands of trials as animals are performing and learning behaviors. And sort of existing dimensionality reduction techniques that are used in practice in neuroscience really aren't equipped to separate out low-dimensional features across these multiple timescales. And the sort of purpose of this talk is to use a generic framework of tensor decomposition as a general purpose framework for actually doing this. And we apply this method with using very minimal assumptions about the data. And as a result, we can apply it off the shelf to a lot of very interesting data sets. So if I have time, I'll talk a little bit about applications to reverse engineering an artificial RNN, recurrent neural network. Also, from Mark Schnitzer's group on maze navigation, and data from Krishna Shenoy's lab in motor learning. But for the first half, I want to sort of take a step back and again talk about like what is the philosophy behind this model and what is it compared to, what are the existing methods in neuroscience. So sort of the simplest method for dealing with multi-trial neural data. So this is the sort of framework that we're going to be working in. We're recording from many neurons over many trials, and each trial is a matrix of neurons by time. And if you're recording, let's say, spiking data, we know that at least a common model for trial-to-trial variability is Poisson noise in the spike generating mechanism. And if really all you think that all that's happening from trial to trial is random variability, a very reasonable thing to do is to just average all of these spike trains and turn it into a PSTH matrix of neurons. And then you can perform principal components analysis on this trial average matrix. So I'm going to call this procedure trial averaged PCA. And this is basically the same equation that I showed you in the first part of the talk, which is that we are approximating the data matrix. So this is X bar because it's averaged across trials. So neuron n at time t is a sum of r principal components, where each component is a low-dimensional representation of the neurons, or a neuron factor, times a low-dimensional representation of the temporal dynamics within trial. So you can think, again, about each of these blue vectors as sort of a bar graph, giving a weight for each neuron, and the red vectors as being a smooth temporal trace. OK, so this actually works quite well in certain contexts and has led to a lot of understanding. Sort of, things that come to mind would be work from Mark Churchland and others in looking at low-dimensional representations of motor dynamics. But this only really works if you're not interested in the trial-to-trial variability. So Byron Yu and others came up with a similar technique called Gaussian process factor analysis. And I'm not going to talk about that explicitly. I'm going to talk about a different model, which I'm going to call trial concatenated PCA. It turns out that these two are extremely similar to each other. And I think this one is simpler to explain. So that's why I'm going to talk about it. But the idea is that rather than averaging across trials, we're going to concatenate all of these matrices into one large matrix. And furthermore, we're going to include an assumption, which is that each spike is sort of a noisy indicator of a neuron's latent firing rate, which is going to smooth in time. So we basically smooth these spike trains with a Gaussian filter. And then we get basically a concatenated and smoothed data matrix. And then we can apply PCA to this data matrix. In GPFA, you don't actually smooth the data. You smooth the latent factors with a Gaussian process prior. But because the model is linear, you can sort of equivalently think about smoothing it in the data space and smoothing it in the latent factor space. But when you fit PCA to this very large, now this is a very large matrix that's very wide. For each trial, you basically get a different set of temporal basis functions. And there's nothing saying that the temporal basis functions on trial 1 are similar to the temporal basis functions on trial 2 or any other trial. So you're sort of separately fitting a different low-dimensional trajectory for each trial. And while this also can be a very useful technique, it doesn't reduce the dimensionality really across trials. And as a result, it hasn't really been used, to my knowledge, to pick out things like learning across many trials, at least yet. So our motivation was, rather than looking just at neuron factors and low-dimensional temporal factors, we wanted to pull out a sort of completely novel low-dimensional representation, which is going to tell us how things are changing on a trial-to-trial basis. And we're going to call these trial factors. And again, the sort of high-level motivation here is that each one of these low-dimensional factors, which our method is going to extract, maps onto different questions that neuroscientists really care about. So the neuron factors give you a sense of what are the cells that fire together and form functional ensembles. The temporal factors tell you how those ensembles are working on every single trial, and actually performing or underlying the behavior that the animal is performing. And then the trial factors tell you how stable those dynamics are over many trials, and whether they're changing to promote learning or homeostasis or some other long-term physiological process. And the idea is actually really embarrassingly simple. So rather than taking your data and putting it into a matrix, it's sort of natural to represent your data as a neurons by time by trials three-dimensional data table. And this is called a third-order tensor. And then we're going to play the same trick as in PCA, which is we're going to try to approximate this tensor as a sum of components. And each component is an outer product of 3 vectors instead of 2 vectors in this case. And mathematically, this is just a very simple extension. The only thing that's changed is that we've added an index to x, and we've added another factor, which is in green here. And these blue vectors give us the neuron factors, the red, the temporal factors, and the green, the across-trial factors. OK, so that's the sort of mathematical. And I should say that this tensor decomposition is actually quite old. It goes back to the 70s. So this tensor decomposition is not new, but overall we call this Surya calls this full model tensor component analysis, or TCA, because it's a very simple extension of principal components analysis. Okay, so that's the mathematical definition of TCA. Now I want to give you a neural circuit sort of interpretation, and this is actually how I think of TCA as a neuroscientist. It turns out that you can show a mathematical equivalence between this existing tensor decomposition model and the idea of trial-to-trial gain modulation, which has been sort of posited as a very important mechanism in cortical computation by Matteo Carandini, Salinas, and others. And so concretely, what do I mean by gain modulation? Well, what I mean is you have a large number of neurons that you record from. So we'll say we record from n neurons, and there's r latent inputs to these neurons. And for simplicity, we'll assume that these are linear synapses and there's no recurrent connectivity between these neurons. Then, and the final assumption is going to be that each one of these inputs has the same waveform on every trial, but only changes in its amplitude gain of these synapses over trials. So for example, this first latent input is increasing in gain over trials. This second one is decreasing, and this third one is increasing then decreasing. And this, this is just synthetic data, but you can imagine that these sorts of trends in amplitude might be a model that underlies things like learning. And that's actually what some of these people think. Okay, so this network model is equivalent to the tensor decomposition mathematical model that I just showed you. Specifically, the neuron factors in this tensor decomposition map onto the synaptic connectivity matrix. So here there's 3 latent inputs and n dimensions. Each one of these is an n-dimensional vector, and there's 3 of them. So that gives sort of the outgoing synaptic weights from each input. Furthermore, the waveform of the latent inputs is given by these temporal basis functions. And finally, the amplitude of the latent signal is given by the trial factor in green, which is the novel low-dimensional component that we're recovering here. Okay, so just to sort of continue to build and motivate the technique, we simulated data from this model network. And then this was sort of the data we got. We added Gaussian noise to show that our technique is at least somewhat robust. It turns out that if you take this simulated data and you apply PCA to it, you don't recover of the true parameters of the model. So what you have to do in order to apply PCA is you have to take this data tensor and flatten it into a matrix. If you flatten it across neurons and apply PCA, you can get a low-dimensional basis for neurons. If you flatten it across time points, you can get a low-dimensional basis for time points. And if you flatten it across trials, you get a low-dimensional basis for across trial dynamics. But each one of these, you sort of run PCA 3 different times, and there's no way of connecting them to each other. And intuitively, that's why this doesn't work. If you TCA, it's actually a sort of remarkable result that you get the exact parameters of the model. So basically, this is saying that this is a statistically identifiable model. And going back to the first part of the talk, the reason PCA fails is sort of by design. It has this rotational ambiguity that I told you. And sort of remarkably, there's a seminal theorem by Kruskal in 1977, which proves that this particular tensor decomposition can recover the ground truth factors, even if they're not orthogonal to each other and even if they have similar magnitude. Whereas PCA only actually recovers the ground truth parameters if you assume they're orthogonal and there's a large eigengap between them. So basically, this model actually picks out features that are, or has a hope of picking out features in your data that are more intrinsically meaningful than PCA. Okay, so as far is that I've introduced a new technique, TCA, that is going to give you separate low-dimensional bases for fast within-trial computations and sort of slower emergent trends that happen over many trials. And despite being a very simple generalization of PCA, it actually has strikingly advantageous theoretical properties. In particular, it doesn't have this rotational ambiguity under very mild conditions. And furthermore, I think using TCA for neuroscience is a particularly good application and sort of ripe for use because there's existing theoretical models of cortical gain modulation that I actually think is an important feature of cortical circuits. So with that, I'm going to sort of focus on 3 applications just to sort of show that this technique can be used in a very general off-the-shelf manner. Yeah? Yeah, so the question is, what are the assumptions in the TCA model, and particularly what are the assumptions on the distribution of the latent factors? So, and also, well, no, it's not just PCA. Okay, so there's a couple things. One, in our application of TCA, while you can add a prior on each of the terms, we're going to apply TCA without any priors. And another way of saying that is there's not going to be any regularization on our TCA model. About the noise. So we're going to fit it with a quadratic loss function. So that's the assumption is that it's Gaussian noise. And that is a similar assumption to PCA. But I guess, yeah, so it's just PCA in the sense that you're solving sort of a similar optimization problem, but it actually has very different theoretical properties. And the main difference is that you're adding this factor in. So it's a simple extension, but it actually is, tensors in general get quite tricky theoretically.
Sure, but there is still some.
Yeah, so
you have some assumption.
No. So there's no distribution. These coefficients can be anything in general. But what I'm saying is you can interpret each coefficient in the across trial factor as being a gain modulation term in this network. Does that make sense? But the gain can be whatever it wants, and the gain can actually be positive or negative in the standard TCA model. Yeah? Where it fails? That's not in my best interest. Where it couldn't where it couldn't recover this. So the yeah, PCA almost never works. When that PCA fails or PCA? Yeah, and in this case, PCA correctly recovers the linear subspace of these factors. It just doesn't recover the actual factors themselves.
For example,
so for this, if you look at the details of this Kruskal paper, basically what he says is as long as the factors, the factors are linearly independent from each other, which is a very loose assumption. But if you have too much linear dependence along the factors, then all bets are off. In practical applications, it doesn't, well, I mean, it depends. I mean, your data might be high dimensional, right? In that case, TCA fails because TCA is assuming you can express it in a low dimensional way. If you just give me noise and I fit TCA to it, it's not going to work. So the way I think about it, so the question is, when does TCA work? If you think that this model is sort of a reasonable characterization of your data or might work, then you should try it. And then if you get a good reconstruction error compared to PCA or other methods, then this is actually a much more compact model with more assumptions built into it than PCA. So if it performs well in terms of reconstruction error relative to those other methods, then this is going to be a better model for you because it has better theoretical properties and it has fewer parameters. So I mean, if it's generated from, if this is the true generative process, and if your data is actually generated from this sort of a network, for example, and you have additive Gaussian noise, and all of these latent factors are linearly independent, then it is guaranteed to be unique. If there's recurrent, then it might still give you something useful. In fact, that's the next example. So maybe I should go to that. So this is sort of the best case scenario for TCA. This is your network. Real neural networks have nonlinear synapses and recurrent dynamics, but yeah. No. So what I'm going to show you now is that in a nonlinear recurrent network, it actually gives you that's quite insightful. So even though the assumptions are technically violated here, and I can't give you like a mathematical theorem that TCA is the right thing to do, we can actually analyze this. We can empirically show that we get something useful out of it. So this is sort of the first application is we're going to look at a model neural network because we can sort of study this we can basically do any experiment we want. We can measure any synapse we want. So this is a very great model system as a proof of principle. And we're going to perform a simple sensory discrimination task where the input cell gets a noisy signal and it's either positive or negative. The job of the network is to, when the input is on average negative, output a negative response. And when the input is on average positive, output a positive response. And the sort of motivation behind this artificial network is that we're trying to solve something analogous to a very classic experiment pioneered by Bill Newsom and others, done in primates as well as in rodents now, where you have basically random dot motion, but if there's coherent motion to the left across this random motion background, the monkey is supposed to respond left. And if there's coherent motion to the right, the monkey is supposed to indicate that response. So this neuroscience experiment is sort of analogous to this in silico model that we're going to reverse engineer. And basically, on each trial, we give the network an input. It gives us some output. But we look at the activity of all of these recurrent model neurons. And then we repeat this across trials. And we collect all of these data matrices and put them into a tensor. And we learn this task using backpropagation through time and gradient descent, which is, we use the PyTorch deep learning framework to do this. But there's sort of many ways that you could imagine performing, training the network to actually do this. But this is just showing that after about 750 trials, the network is performing basically at 100% accuracy. So how did it start from totally random weights to something that actually performs this task? And interestingly, if you look at individual cells, you get the sense that this gain modulation hypothesis, which is what TCA is sort of fitting to the data, actually is going to work quite well. So if you look at, this is one particular cell, and these two grayed out traces are early before training, versus after training, the black trace is much higher and the red trace is much lower. And they've sort of been scaled comparably. And likewise, you can find cells that have the opposite behavior that respond by increasing their activity when the input is negative and decreasing their activity when the input is positive. In fact, if you look at a scree plot showing the improvement in fit as you increase the number of components in a TCA model, you can explain the vast majority of variance, around 75%, with just a single component. And you can see from this that this is still a pretty noisy network, and you can get 75% of the variance with TCA model. And this is even though there are nonlinear synapses and recurrent connectivity. So there isn't necessarily a mathematical guarantee that this should work, but empirically it sort of does. Okay, so we have a single component model. So this is an our model has basically picked out an extremely simple picture of the data. And what does it tell us? So by looking at, first we're going to look at this neuron factor picked out by TCA. And we're going to sort the cells from largest to smallest on this factor. And basically this recovers cells that respond positively to a positive input and cells that respond negatively to a positive input, and vice versa if the input is switched. So this basically picks out a spectrum of cell types. The temporal factor shows that all the cells are integrating evidence at a sort of similar time scale within trials, and eventually they reach a plateau. And the across-trial factor is interesting and has the most going on. And remember, this is the one that you really can't get without TCA. And what this shows you is that at the beginning, on the plus trials and minus trials, the output of the network is very similar. But as the network learns the task, the plus trials and minus trials become separated by basically a gain term that's positive versus negative. So here, the trial factor is more or less separating the trials where the network responds that it's a plus and the network responds that it's a minus. So you get trial by trial decoding of what the network is doing. And furthermore, you get a learning time course from this. That make sense? Well, so yeah, I don't have a actually shows that, but basically my argument would be if the time course was not shared, you would need multiple components. So TCA is sort of saying that I can describe all of my neurons as having this time scale and this gain across trials. And you'll get 75% of the variance with this. So yeah, they might not all be exactly, you can definitely take a microscope to this thing, but at a high level, this is a pretty good model of what's going on. Okay, so, and then, I guess I have time. So another thing, you can take this neuron factor and resort the connectivity matrix. Because this is an in silico model, we can look at the synaptic connectome of this model. And at first, it looks like complete noise. But if you resort the connections by the neuron factor extracted by TCA, you can see that there's sort of competitive inhibition between the plus cells and the minus cells. And this basically leads us to a very simple description of what's going on in the network, which is basically a winner-take-all connectivity. And if you study sort of integration networks, this result actually should not be surprising to you. We sort of knew that this is the computational mechanism that the network should converge to based on previous work by Sebastian Sung and many others. But the fact that we're able to recover this in a pretty straightforward manner is just good validation that TCA can actually point us in the right direction once we start to reverse engineer networks that are more complicated and doing tasks that we don't fully have a handle on. Okay, so that's the first application. So now let's talk about experimental data. This is now work done in collaboration with Tony Kim and Forey Wang and Mark Schnitzer's lab. So they have a task, and I mean, you don't have to actually pay attention to the details of this too much. But basically, mice start on either the west or east arm, and the opposing arm is blocked in a 4-arm maze. So on every trial, the mouse has a binary decision whether to go to the north arm or south arm. And also, there's a binary outcome, which is whether the mouse receives a reward or receives an error. And they did large-scale calcium imaging in mice over multiple days while they sort of navigated this maze. So this is a video that should show behavior. So the mouse is starting here. He, in this case, goes there. He starts licking because he got a reward on that trial. And now he starts on this other trial. And meanwhile, they're using a miniscope from Inscopics that records activity, single photon calcium imaging in principal cells. Okay, so this is medial prefrontal cortex, and one of the first questions that we wanted to look at was, well, how good does this TCA gain modulation model actually work in this data set? And if you look at individual cells, again, you sort of get the same sense that this gain hypothesis actually does a pretty good job. So here on the left, this is one cell in raw data, and I'm showing the average fluorescence for that cell on east starts, plus or minus some standard deviation, and west starts. So this cell is selective for the east starts in the maze. and this cell down here is selected for the west starts. When you apply TCA, here we're doing TCA with 15 components, but we could even probably do less, more like 10 components. The reconstruction of TCA also matches this, and it's perhaps not too surprising because really there's just the difference between these conditions is a gain modulation. This is perhaps the killer figure for this application, which is that we compared TCA as well as non-negative TCA. So non-negative TCA is just NMF, non-negative matrix factorization, but extended to tensors. It's also a very straightforward extension. So here we have, and we're comparing the TCA models to trial averaged PCA and trial concatenated PCA. The trial averaged PCA obviously is missing, we're averaging over all trials, so we're missing any particular coding that the cells have for versus west starts and other task variables. So that ends up flattening out around just under 0.6 error. Whereas TCA and non-negative TCA can go much lower than that. And on the x-axis here, we're looking at the number of parameters in these models. So you want to sort of be more to the left to have a larger dimensionality reduction, and lower so that you have a lower error. So you can see that trial concatenated PCA, which remember is sort of similar to GPFA, actually performs pretty similarly to TCA and TCA. When you go up to maybe 20 components, it starts to outperform a little bit, but I'm convinced that this is overfitting anyway. But the important thing here is that when you look early on, they get similar model error, but trial concatenated PCA requires basically 2 orders of magnitude more parameters to fit this data. So TCA and non-negative TCA do a very good job of fitting the real data, and they do it with many fewer parameters. And as a result, you get those nice theoretical properties of identifiability, like I mentioned. OK, so what do the TCA factors actually pick out in this case? Again, because we don't constrain them to be orthogonal, we can actually sort of directly interpret the results of TCA in terms of functional cell ensembles, canonical firing rates for those cell ensembles, and the pattern of activity across trials for that population of neurons. So I'm just showing you 8 factors here. The top 2 are selective for east versus west starts. And you can see that these neurons are active at the beginning of each trial. And furthermore, they're sort of disjoint sets. There aren't too many neurons that code for both east and west. These factors are perhaps more interesting. This one is independent. It's active on every single trial. And it fires around the midpoint. This was an unexpected result, although in retrospect maybe should have been expected. Basically, within each day, there's a decay in the fluorescence of many cells. And this could be, if it's an uninteresting explanation of this, is that it's fluor bleaching. More interestingly, it could be something like a thirst or motivation signal within prefrontal cortex. But the point is that we weren't necessarily expecting to pick this out, but TCA picks it out, and furthermore, it picks it out in its own factor. And then the final 4 also code for interesting things like whether the mouse ends up in the north arm or the south arm, and whether the mouse gets rewarded or the mouse had an error on that trial. And interestingly, if you do trial concatenated PCA on this, try to look at the trial factors, you get all sorts of mixtures. So the real result here is that when you look on the across trial factor, you get nice, each factor sort of codes for one task variable at a time. Here, if you look at the top 6 principal components, you get this within day activity sort of in here, so down here, and sprinkled in up here. The top factors sort of maybe codes for east versus west, but there's all sorts of ugly mixtures here that aren't necessarily easy to pull out. And again, this is just basically because of the orthogonality assumption that's into PCA, and we don't have in tensor component analysis. OK, so that's actually perfect. That can be done in 5 minutes, and then we can do questions or exercises. The last application comes from Krishna Shenoy's Krishna Shenoy's lab, data collected by Saurabh Vyas. And here we were interested in studying how a monkey learns to use a BMI. So here the monkey's controlling a computer cursor using by sort of imagined motion in motor cortex. So there's a Kalman filter decoder that moves your virtual computer cursor on the screen. And initially, I'm showing just 45 center outreaches from the center of the screen to this 45-degree target. Initially, the monkey does quite well because he's done this task before. And then what Saurabh did was he perturbed the BMI so that when the monkey tried to execute a 45-degree reach, the cursor went off at a 30-degree angle. And the task for the monkey was it had to basically learn to adapt and compensate for this perturbation and make more direct reaches. And within a single session, the monkey more or less was able to do that, or at least get a partial recovery. OK, so jumping to the end, if you apply tensor component analysis to the neural activity on the 45-degree reaches, a 3-component model does pretty well. And it sort of captures interesting things. So first, this first component we call an early component, which basically is on all trials, is active at the early part of each trial. And this captures the neural dynamics that execute the 45-degree reaches at the beginning of the session. And interestingly, after the perturbation comes on, these dynamics do not extinguish or die away. Even though the perturbation, they're actually doing the wrong thing. They're pushing you 30 degrees off because of the perturbation. So rather than actually extinguishing these early components, what the monkey does is it adds 2 more components. One is a corrective component that immediately comes on, and the other is a slower or learned component, is what we call it, which comes later and it more slowly ramps up. And just to give you sort of an idea of what I'm talking about here, in the initial reaches, the red factor, or the early component, is the TCA factor that's explaining the majority of the variance in the data. After the perturbation, the red factor is still most active at the beginning, so the monkey sort of hasn't learned to extinguish these maladaptive dynamics. And instead, he makes a cursor movement upwards, and then he makes a corrective movement, a late-term corrective movement within the trial. And within that, this second TCA component is explaining the most variance in the data. And then finally, by the end of the session, you see the appearance of this learned component, or the green component, and that sort of helps the monkey make more direct reaches. So the idea is that the maladaptive dynamics do not go away. The monkey sort of brings on sort of extra cognitive effort to correct the early incorrect cursor movements that are created by this component. And then over the course of the session, a more stable component that actually has better performance emerges. And this picture largely replicates when you run TCA on each of the reach angles. So this is showing 45-degree reaches. Here, this is just showing the tuning curves across all 8 reach angles. And you see again that the early component is off, has an error to the left. The corrective component is sort of mirroring this and is off to the right to sort of impose this late-term correction. And the learned component is not significantly different than 0. So this is sort of, in some cases at least, seems to be neural dynamics that are actually performing the correct task. OK. So that's basically it. Just to summarize, I'll say that there's growing interest in doing these large-scale neural recordings, but also doing them over very long time periods while animals are performing naturalistic behaviors or learning complicated tasks. And the idea of this work was that we can use tensor component analysis off the shelf on these data with very minimal assumptions. I've shown you that TCA has nice theoretical properties, and these are reviewed in many papers. One really nice review is done by Tammy Kolda, who's my advisor at Sandia in 2009. This is sort of the canonical review of tensor decomposition methods. But even though this is a nice existing technique and has very well-characterized theoretical properties, it also ends up being related to existing ideas within neuroscience and the idea of gain modulation. And specifically, the nice theoretical properties proved by Kruskal and others basically show that identify this model and pull out meaningful characteristics of our data to a greater extent than PCA. Furthermore, as I sort of mentioned in the first part of the talk, all these models can sort of be seen as, especially when you add non-negativity constraints, you can think of these models as being somewhere in between PCA and clustering. So you can sort of reveal discrete populations of cells with discrete activity, trial-to-trial activity modulations. And this is sort of a soft or pseudo-clustering of your data. So TCA can sort of pull that out if that's what best describes your data. But you can also pull out more gradual trends in your data as like slow learning after perturbation or a continuous spectrum of cell types. So TCA is a really nice technique that lets the data sort of speak for itself and is sort of natural in many of these multi-trial, large-scale neural experiments. And with that, we can do a few exercises. I have some code for fitting these tensor models, and there's also other packages out there that I'm happy to talk to you about. And I just want to thank all my really great collaborators on this one more time, and thank you all for listening. Optimization-wise, it's the same optimization algorithm actually. So I didn't talk about optimization at all, but you can use an alternating minimization like NMF, and you basically that involves flattening the tensor into a matrix across different dimensions. So extending it to higher dimensions algorithmically is no challenge. The only problem is that as you go to higher and higher dimensions, your data can actually get very large because there's this multiplicative effect, right? But yeah, if you have an interesting idea or application of adding more dimensions, that I would love to hear that. One thing that you have to be a little bit careful about is whether this, so in the case of dimensions, we have this nice interpretation of TCA as gain modulation, and I actually think that's pretty important because if you go to more dimensions than that, it actually could be quite a constrained model, and you have to think sort of carefully about how do you interpret higher order tensor models. Yeah, so this slide here, so DPCA is basically was created to solve this problem for PCA. But there are many differences, and I don't know how much you want to go into them. But for example, DPCA really does not give you this third low dimensional factor across trials. You're still basically doing trial concatenated PCA, so you have many parameters in your model at a different full trajectory for every trial, which actually might not be enough dimensionality reduction if you have a large enough data set. Another thing with demixed PCA is it has the same rotational ambiguity problem within each subspace. So if you fit demixed PCA to separate east and west, north and south, et cetera, within the north-south subspace, for example, you can play the same rotational tricks. So basically, your intuition for PCA, some things actually do extend over to demixed PCA. That being said, I think demixed PCA is a pretty interesting technique. There's just different strengths and weaknesses with using that versus TCA. One thing that's nice about TCA is you don't have to actually know, like for example, this factor here we didn't know about, but TCA nevertheless sort of picked it out. And that's sort of the advantage of using a fully unsupervised. But in cases where you know exactly what you're looking for, using demixed PCA might be a more targeted tool. Yeah. So okay. So the question is, have I thought about adding more constraints to this model to reduce the number of parameters even further? So one way of adding simplicity to sort of restate your question, a further way of adding or decreasing the number of effective free parameters in this model would be to add a sparsity constraint. and use a sort of sparse coding framework. My understanding with most of the sparse coding work is that there's a little bit of a difference because you usually have a large dictionary and then you just sort of pick a sparse subset of that dictionary. Whereas here, in practice, you have a very small dictionary and there's sort of a dense selection method. I think, yeah, but regardless, you can add sparsity penalties to TCA. You could probably if you have an idea of what your basis functions should be, for example, you could use a series of like cosine basis functions, and you could perhaps work to add more assumptions into your model that way. And I think that it wouldn't be too hard to connect this to things like sparse coding. And actually, I'm sure that people have done that in other non-neuroscience contexts if you did a Google Scholar search. Does that answer your question? Yeah. Yeah. I mean, the motivation behind what I've done so far is to try to keep things as simple and as general as possible. But I agree that this opens up the door to very interesting future work on more specific and constrained models.